When most people think of a term such as artificial cloning, they typically would think of something like this, when in reality it is closer to this. Hello, my name is Richard Swanson. I'm 15 years old and a 10th grade homeschooler. This is my 11th year in 4-H and my 9th presentation given at the county level. I am a member of the Valley Green Acres Horse 4-H Club. The title of my presentation is Artificial Cloning. I'm going to be talking to you about the definitions of cloning-related terms, the history of artificial cloning, and the various methods of artificial cloning. Neither plant, fungus, or human cloning fall under the scope of this presentation. Only animal cloning does. And, in case you were wondering, this was a Przewalski horse cloned just last year. The definitions of cloning-related terms. The definition of clone is as follows, quote, a group of organisms derived from a single organism by various types of asexual reproduction, end quote. From this definition, it should be immediately obvious as to why the title of this presentation is Artificial Cloning and not merely cloning, because I will not be covering natural forms of cloning in this presentation, only in artificial forms. Before we jump into the history of cloning, we first must understand what DNA is. DNA is that twisted ladder-like structure in the nucleus of just about all your cells. It is made up of the 4 base pairs A, T, G, and C, and a sugar phosphate backbone. These 4 genetic letters, A, T, G, and C, make up 3-letter words called codons, which code for 20 different amino acids. The specific arrangement of amino acids is linked together to form proteins. The length of DNA that codes for a protein is called a gene. The definition of chromosome is as follows, quote, any of the several thread-like bodies consisting of chromatin that are found in the cell nucleus and carry the genes in linear order. The definition of nucleus is as follows, quote, a dense organelle present in most eukaryotic cells, typically a single rounded structure bound by a double membrane containing the genetic material, end quote. The definition of somatic cell is, quote, any living cell other than the reproductive cell. Any cell of a living organism other than the reproductive cells, end quote. The definition of epigenetics is, quote, the study of changes in an organism caused by modification of gene expression rather than the alteration of the genetic code itself, end quote. In other words, epigenetics control which genes are on at any given time without alterations to the genetic code, to the DNA. For example, histones are a protein that DNA is coiled about for compact storage. They're sort of like spools in that sense. But what is fascinating about them is that they can control gene expression by how tightly the DNA is wound about them. If it is wound tightly, the DNA cannot be read and transcribed into protein, and vice versa. Even more fascinating is that when the DNA is copied in cellular division, these epigenetic changes are passed on to the daughter cells. I would love to elaborate on the topic, but it would take a presentation in and of itself to even partially explain epigenetics. All that I can add quickly is that epigenetics can be influenced by the environment. The zona pellucida. The zona pellucida is that thick transparent membrane surrounding a mammalian ovum before implantation. The history of artificial cloning. As you will recall, the definition of clone is, quote, a group of organisms derived from a single individual by various types of asexual reproduction, end quote. From this definition, it should be immediately obvious that cloning is far more commonplace than commonly thought, covering everything from potatoes to occasionally parthenogenic lizards and E. coli bacteria, all of which do not fall under the scope of this presentation. It is important to note that identical twins are clones of one another caused by the natural division of an embryo early in development. This is possible because in early embryonic development, all the cells are undifferentiated and can turn into any cell in the body. Although it is far less common, the exact opposite occasionally occurs, when one embryo absorbs another embryo. This results in a chimera, quote, an organism containing a mixture of genetically different tissues formed by a process such as fusion of early embryos, grafting, or mutation. The first example of artificial cloning was demonstrated by German embryologist Hans Spemann, when in 1902 he split a two-celled salamander embryo, which successfully developed into two separate salamanders. In 1952, R. Briggs and T.J. King successfully devised a method of nuclear transfer, that is, the transfer of the nucleus of one cell into a cell that has had its own nucleus removed, in which they enucleated, that is, removed the nucleus of, an unfertilized frog egg, and using a micropipette, injected the nucleus from a frog embryo. J.B. Gordon and R. Lasky demonstrated that if the nuclear donor was a blastula cell, a very young embryonic cell, the combined cell developed into a salamander 80% of the time, whereas if the donor cell was from an intestinal cell, the success rate was far lower. In 1975, Derek Bromhall transferred the nucleus of rabbit embryo into an enucleated rabbit egg. This was the first successful attempt at nuclear transfer in mammals. Bromhall did not place the clone into a surrogate mother to see if it would develop successfully. In 1984, Steen Willitson fused a cell taken from an 8-celled sheep embryo with an enucleated sheep egg. This method resulted in the birth of 3 live lambs. In 1987, a method very similar to this resulted in the birth of 2 calves, respectively named Fusion and Copy. You have probably noticed that all the previous listed times when a mammal was cloned, the donor nucleus came from embryos. In 1996, Ian Wilmot and Keith Campbell pioneered a new technique using donor nuclei from laboratorily cultured sheep cells. What is significant about this new development is that it is easier to genetically engineer cultured cells than it is to engineer embryos. 2 lambs were created by this method. They were named respectively Megan and Morag. Also in 1996, and also created by Ian Wilmot and Keith Campbell, was arguably the most famous clone in history, Dolly. Dolly was created by transferring the nucleus from a cell taken from an adult Finnish Dorset's udder into an enucleated sheep egg. This is particularly significant because in adult cells, the genes that build the proteins necessary to create an embryo are turned off. This was the first documented time when the mammalian cells had been reprogrammed back to the embryonic state. It took them 277 embryos to get Dolly. This equates to a roughly 0.4% efficiency rate. Some people claim that Dolly's genome was pre-aged because the cell line that she was produced from came from a middle-aged sheep. Their main evidence of this claim is that Dolly died at the comparatively young age of 7. However, Dolly did not die of old age. She was euthanized due to a cancerous retrovirus. And 4 other sheep were cloned from the same cell line as Dolly, and they all lived to the ripe old age of 10. Following Dolly, a large number of different species have been cloned, everything from mice to monkeys, and the occasional endangered species, and a clone of an extinct ibex in 2009. It died due to lung defects shortly after birth, though. Interestingly, 2 of the endangered species cloned were born just last year, the Przewalski's horse and the black-footed ferret. Even more interestingly is that they're cloned in collaboration of the same 3 organizations: the San Diego Frozen Zoo, Revive and Restore, and Viagen Pets and Equine, where you can clone your favorite dog, cat, or horse for a fee. Various methods of artificial cloning. The only methods of artificial cloning that I am aware of are artificial twinning and nuclear transfer. Artificial twinning is by far the oldest method of artificial cloning. There are three methods of artificial cloning: mechanical, chemical, and a combination thereof. The earliest method of artificial twinning involved tying a hair about a two-celled salamander embryo and then pulling the knot tight. Unfortunately, this doesn't work with mammalian embryos due to their small size when compared to amphibian embryos. Nowadays, we have more advanced techniques, such as non-violently chopping up 8-celled embryos into 4 equal parts. They found that this is easier than chopping up 4-celled embryos into 4 equal parts. Another method is to suck off a cell or two with a micropipette. In the chemical method, the zona pellucida is first dissolved by a mildly acidic solution, and then the embryo is separated by trypsin. And well, that's just about it. In the combination thereof, the zona pellucida is first dissolved, and then the chosen mechanical method is carried on, whether it be chopping or sucking. Nuclear transfer can be split into two subgroups: somatic cell nuclear transfer and embryonic cell nuclear transfer. Of the two, embryonic cell nuclear transfer is the easier and older method. Between the two subgroups, the methods of moving the nuclei are just about identical. It is the success rates of reprogramming the nuclei that are different. Nuclear reprogramming is the resetting of the cell back to the stage where it is now interested in making a body again. For example, the differences between embryonic cells and other embryonic cells aren't as different as, let's say, embryonic cells and skin cells. So, needless to say, the skin cells need to be reprogrammed back into embryonic cells. This is accomplished by the recipient egg, not by any fancy laboratory method, although nowadays it can be. The success rate of embryonic cell nuclear transfer are up to 6 times more efficient than somatic cell nuclear transfer. A problem that is common to both of the subgroups is when the nucleus is in one stage of the cell cycle and the recipient egg is in another stage of the cell cycle, such as if the nucleus is dividing and the egg is sitting tight. This understandably causes some problems, most notably death. A common method of synchronizing the cell cycle is to starve the cell for a period of time. The first step in nuclear transfer is to enucleate an egg. This can be done by a variety of methods, such as the destruction of the pre-existing nucleus with ultraviolet light, sucking out the nucleus with a micropipette, chopping the egg in half to get a side, and chopping the egg in half to get a side without the nucleus. Step 2 is injecting the nucleus. The methods of accomplishing this are even more varied, covering everything from fusing the donor cell to the side of the egg with electric shocks, injecting a nucleus with a micropipette, adding a nucleus to the egg with electric shocks, and amalgamating a nucleus to the egg with an inactivated virus. Following this, the reconstructed embryo is shocked multiple times to restart cell division. The final step is to implant the embryo in a foster mother so that it can develop to full term. It is important to note that animals born by nuclear transfer are more likely to suffer from birth defects and large offspring syndrome than naturally reproduced animals. In summary, I have told you about the definitions of cloning-related terms, such as epigenetics, the history of artificial cloning, such as the creation of Dolly, and the various methods of artificial cloning, such as embryonic cell nuclear transfer. My sources of information have been academicoup.com, creation.com, US patent number U.S. patent number 7232938B2. The International Journal of Developmental Biology, www.newport.org, U.S. patent number 6603059B1, U.S. patent US 20130007904A1, World Book 2002, the Random House College Dictionary, ViagenPets.com, and the sixth edition of Integrated Zoology by Hickman, and Roberts, and Biochemistry by Stryer. This concludes my presentation. Thank you for listening.